there everyone! In this video we'll be exploring molecular mass spectrometry. In addition to elemental analysis, like we were just looking at previously, mass spectrometry is very widely used today to study the structures of inorganic, organic, and biological molecules. It's also capable of determining both the qualitative and quantitative composition of complex mixtures. So the exact same principles that we saw previously apply, but now we want to do molecular analysis. So we don't want to bust up the molecule like we were before. Instead, we use different ionization techniques to produce molecular ions. Like you can see in this mass spectrum of ethylbenzene, the peak for the molecular ion occurs at 106, mass to charge ratio of 106. Now, relaxation of some of these excited ions results in fragmentation, which is resulting in these peaks with lower masses. As you can see, this base peak here at m over z of 91. And the base peak is just the peak with the highest relative abundance, and then all other peaks in the spectrum are normalized to that. The starting point for mass spectrometric analysis is the formation of ions. So we're going to focus on 3 different ion sources. As you can see, there are many, but we're going to focus on 3. So there's electron ionization, which we're going to look at, electrospray ionization, and matrix-assisted laser desorption ionization, or MALDI. So these ion sources are classified into 2 different types, hard and soft. Hard sources produce more fragmentation, which is good for example, determination of functional groups, and electron ionization is a type of hard source. The second is soft. So soft sources produce very little fragmentation, which is good for accurate molecular mass information, for example. And the second 2, ESI and MALDI, are both soft ionization techniques. So the first ionization technique we're going to look at is electron ionization, or EI. And this is when a sample is brought to very high temperature in order to vaporize it. And then you can see it enters into the system here. And it's ionized, or the sample is ionized by bombardment with this beam of electrons, as you can see here. That's coming down perpendicular to the sample. And as a result, extensive fragmentation occurs, which results in these things called fragment ions. However, the molecular ion is often not preserved. So usually what you end up with is only these fragment ions. This happens sometimes, sometimes not. So this source can be good for compound identification as long as the molecular ion peak remains present. So the pros are that it's convenient to use, and like I said, good for compound identification with the caveat that the molecular ion is preserved. However, the cons are that high temperatures that are needed to volatilize the sample can cause thermal degradation. The other thing is that only molecular masses less than 10 to the 3 Daltons can be used in this ionization technique. So next up is MALDI, or matrix-assisted laser desorption ionization. And this desorption ionization method is good for thermally delicate samples, for example, biochemical species and species with molecular masses greater than 100,000 Daltons. So a much wider range of molecules are able to be probed using this technique. And it can also obtain accurate molecular mass information since the spectra only contain the molecular ion peak. So because it's a soft ionization technique, you're not producing all of these fragment ions, and you basically only get the molecular ion peak. So in this technique, you first mix the analyte with a solid or liquid matrix and drop it onto a metal plate, as you can see down here. And then a pulsed laser strikes the sample, causing desorption of this plume of matrix, analyte, and the ionized analyte molecules. And this can then be filtered out and sent to the mass analyzer. So the final ionization technique we'll look at is electrospray ionization, or ESI. And it's probably the most widely used technique, especially for studying biomolecules and polymers. And it takes place under atmospheric pressure and temperature. And the sample is first dissolved in a volatile solvent. And it is then pumped through this charged capillary needle that you see here. So this charged needle results in a charged spray of fine droplets that then pass into this capillary here. And as these analyte ions travel through the capillary, the solvent evaporates and forces the droplets apart into a whole bunch of smaller droplets. And then these smaller droplets undergo this again and again, and it's repeated until there's no solvent left. Now, the pros of this technique are that very little fragmentation occurs, and so you end up with very simple spectra. And because of this multiple droplets forcing apart into more droplets, you end up with multiply charged ions, which means they have very small mass M/Z ratios. And this means that detection by quadrupole instruments is possible. It's also readily adapted to direct sample introduction from other instruments, for example, HPLC or high-pressure liquid chromatography or CE, etc., etc. We'll get back to these later in the course. The con, however, is that no fragmentation means that it's very difficult to determine molecular structure using this method because you're only ending up with basically one peak from the molecular ion.